Today we're going to be talking about transcription. And transcription is the process by which we take the information in DNA and convert it into RNA. So we're going to go ahead and we're going to start with DNA. And this is double-stranded DNA, modeled double-stranded DNA. And the first thing that's going to happen here is RNA polymerase, which is an enzyme, is going to scan the DNA for a sequence of bases that tell it where to actually attach and start the transcription process. This sequence of bases that tells the RNA polymerase where to start is called the promoter. So we're going to look along the RNA for a sequence of bases called the promoter that tells the RNA polymerase where to start. Once the RNA polymerase finds the promoter, it's going to attach to the promoter, it's going to unwind the DNA, and it's then going to begin to separate the DNA and start the transcription process. So let's go ahead and start there with our diagram. So I'm going to draw my double-stranded DNA. There it is. And this is going to be the 3' end of my DNA. And the other side is going to be the 5' end. And because DNA is anti-parallel, if this is 3', this must be 5', and then 3' on the other side. So this is my double-stranded DNA. Let's say the promoter is located right here. RNA polymerase is attached to the DNA, and we will show that right here. This is going to be the enzyme, RNA polymerase. And it's going to attach, it's going to separate the two DNA strands, and it's going to begin to make RNA. So let's go ahead and begin to show that. So the next step, we're going to take one of these strands, and for convenience, let's go ahead and use the top strand, because not both strands do not get copied. Only one of the two strands actually gets copied in this process. So we'll take the top strand, the 3' end, the 5' end. Here's my RNA polymerase. And we're going to start actually making RNA right here. So this is going to represent my RNA that I'm making. And we're going to make it in this direction. Now, just like DNA replication, transcription is going to proceed in the 5' to 3' direction. We're not going to read it in that direction, but we're actually going to make the RNA from 5' end to 3' end. So we're going to start here. This is where the RNA began to be made. So that's going to be the 5' end, and over here is the 3' end, because we're making it 5' to 3'. And again, these two strands, the DNA strand and the RNA strand that we're making, have to be anti-parallel. Now the DNA strand that we're actually using is going to be called the template strand. And it contains the information that we want. And we're going to make a complementary copy of RNA. So this DNA, and here's the complementary copy, the RNA. Now the RNA is not finished yet, so we're going to call this pre-messenger RNA, because it's not finished. It has to go through some processing still. Okay, now the next step, once we're finished making this messenger RNA, and this process continues until the RNA polymerase actually finds a sequence of bases called the termination site. And once it reaches that termination site, RNA polymerase releases the RNA, and that's where the next step is actually going to begin. So let's go ahead and draw that. So here's the messenger RNA. Let's call this the 5' end and the 3' end. And the messenger RNA at this point is actually going to have special sequences attached to the 5' end and the 3' end. So the first thing that's going to happen is we're going to attach a GTP molecule, a modified GTP molecule to the 5' end. And this is going to be referred to as the 5' cap. And it's going to help protect the messenger RNA when it gets into the cytoplasm, because there's a lot of enzymes that want to break this messenger RNA down very quickly. It's also going to allow this messenger RNA, which is currently right now in the nucleus, to leave the nucleus through the nuclear pore and go into the cytoplasm. Now on the 3' end we have a sequence of nucleotides also called the poly A tail. And this is just a sequence of many adenine nucleotides, approximately 200 or so. So this is going to be referred to as the poly A tail. And it's usually around 200 nucleotides long. And again, it serves the same function, to allow this messenger RNA to actually leave the nucleus and to help protect it temporarily when it gets into the cytoplasm. Now this piece of pre-messenger RNA is actually made up of several regions. All right, we'll call this E1 for exon 1. Exons are actually coding regions of the RNA. These are actually regions of the RNA that are actually used to make our finished protein. This is going to be an intron. This is an intervening sequence. This is a piece of RNA that's not real, doesn't contain any information to make the protein. So ultimately we're going to discard these introns because we don't need them. This will be exon 2, intron 2, and finally exon 3. And there could be many more introns and exons than this, but for simplicity, let's just take it this far. Okay. Now the next step, once we've got this done, is going to be called RNA splicing. And to splice is actually to cut out and remove. So using an enzyme, we're going to cut this piece of RNA between the introns and the exons, and then we're going to remove the introns and connect up the actual coding regions, the exons, to make our finished RNA. So I'm going to go ahead and get rid of these introns. We're going to remove those, and get rid of them, break them down, and we're going to show our finished messenger RNA. Again, the caps are still there, the GTP still there, the poly A tail still there, and let's connect up our exons. So this would be exon 1, exon 2, and exon 3. So our finished RNA is only made up of coding regions called exons that can be used to make a protein. Now, this is very important here, RNA splicing, because you can see that if we arrange these exons in different orders, I'll actually be able to create different proteins later on. And this saves the cell a lot of space. In a sense, we can take one bit of information here and make several different proteins by arranging these exons in different orders. And the body does this all the time. The cell does this all the time. Let's go ahead and label this as RNA. This is our finished transcript or mature RNA. This RNA will now leave the nucleus through the nuclear pore, go into the cytoplasm, where the next process will begin called translation or protein synthesis.